Hello, welcome to the first concept overview video. This will be for the alterations of cell function. Because it's the first one, I'm going to walk you through how to find the material that you need. So I'm going to share my screen so you can see. We're going to go to the home page of our class. This is what that looks like. The way to navigate through our class is to go to modules. Then we're in the first module. You'll have to scroll down a little bit to come to module 1. We want alterations of cell function. And each time that there's a concept overview, you'll find a link for it here. Hopefully you've gotten to this link perhaps through an email. And to get to the overview and lecture notes, you'll scroll down a little further, excuse me, to where it says lecture. And you can find these lecture notes in two forms. If you're going to download and take notes digitally, go ahead and use the Word file. But if you're going to print it out, it's a sure bet to print out the PDF file, because that way the page numbering stays consistent. You don't have to worry about weird blank pages, which sometimes happens with the Word document. Okay, let me show you the Word document in same material. It appears on the PDF. This first page is what we're going to use for the overview, and I've included it in the same file so that you can see that same information. We'll touch on very briefly on page 4 of our lecture notes. But since we're going to go over it in more detail now, hopefully you'll have a richer understanding, or will have been reminded of some things that you already knew before class. So let's go to our PowerPoint. Oh, let me go back to this for a second. The PowerPoint files will always be available to you. We don't recommend printing them out. They're not necessary, but some of you are very detail oriented and want to see the details. We assure you there's no new information that's in the slides that's not in the notes. So the notes are an easier way of taking notes. And we want to streamline this as much as possible for you. So PowerPoints are there, but you don't have to print them out. So I'm going to use the PowerPoint to guide the lecture. All right, so here we are, the concept overview of cell function. You all know human beings are living organisms, and all living organisms are made of cells. So we're going to talk about what cells are, what they're made of. This is a cartoon that Dr. Doig produced that captures all the important stuff that we need to do in a cell. Let's start at the outside. There's a plasma membrane. You'll hear this multiply called plasma membrane, cell membrane, phospholipid bilayer. It all means the same thing. It's a double layer around the outside. The phospholipids, the lipid tails, face in towards each other. Then the phosphate heads are on the outside and on the inside of the cell. Embedded in that phospholipid bilayer, there are some proteins. Showing 2 of them here, a sodium-potassium pump, which we're sure you've heard of before, and a calcium pump. This one is less famous. They do similar jobs. They're both involved in pumping ions against their concentration gradients. That is, we're pumping ions to where we already have a lot of that particular ion, and we're trying to push more in. That's why we call it a pump. We're trying to push more in or out of the cell. That's what the term pump refers to. This is in contrast to, say, a channel that would allow ions to pass through down their concentration gradient. That's just open all the time. This, we're going to have to pay it because we're pushing it, and that's going to take some energy, and so we have to use something to pay it. And as you'll see, that's ATP. All right, so sodium, potassium, calcium pump. So where does this ATP come from that we need to pay these things? There's an organelle known as mitochondria. Mitochondrion, singular, plural is mitochondria. They're fantastic little organelles. They're variously called. The powerhouse of the cell, which I never found informative as a student. Just think in terms of it takes energy in one form and converts it to another form, and it's the really usable form, and that's the adenosine triphosphate, the ATP molecules that we use to pay for everything all over the cell. We'll talk about that in more detail later. Let's see, we are eukaryotic organisms, so most of our cells have a nucleus. There are a couple of exceptions, but the vast majority of our cells have a nucleus containing the DNA. And you'll remember what DNA is for largely is for including the instructions for making proteins. So we do transcription inside the nucleus that sends out RNA from the nucleus to connect with ribosomes. Those are those small organelles that are used to translate, to use the RNA information to tell us what amino acids to bring in to make a long protein or a long polypeptide, which we then fold up and make protein. So that's the job of the ribosomes. Many of these ribosomes are studded on the outside of a structure called the endoplasmic reticulum. Under a microscope, the endoplasmic reticulum with the ribosomes stuck on it, before anybody knew what these things did, it looked rough. So it's called the rough endoplasmic reticulum. I'm not kidding, that's why it's called that. We know more about it now, but we still keep that name. So the rough ER is used for making proteins, packaging proteins, and helping to ship proteins out. Lysosomes. These are little organelles that contain digestive enzymes. So lyse means to cut or to split, and some means body. So these are the little bodies that are used in digestion, in breaking things down. So I think of them as little stomachs of the cell. And we will see in class what happens when we are not able to pay all of these things. Let's talk a little bit more detail about things going right. All right, so ATP production. This is what we need the oxygen for. It is true that we can make a tiny amount of ATP without oxygen, but to make the amount of ATP that we need to stay alive, we absolutely require the presence of oxygen. That is what the oxygen is for. That's the only thing that we need the oxygen for. I'm not making it up. There is a respiratory system in the body. We breathe in, we breathe out. We breathe in, we take the oxygen from that gas that we breathe in. That's the only gas we need. We take the oxygen, and where we need it is inside the mitochondria. We'll talk in a little bit more detail later, but that is what the oxygen is for. It's absolutely required for something called the electron transport chain. In addition to the oxygen, we actually need a source of fuel. Here we're representing that with a G for glucose, but these can be other energy-rich molecules, as we'll see later in the semester. But you probably learned about it as glucose. It's not just glucose. Cells can use other forms of fuel, but glucose is largely used, and it's probably what you learned. So you use that fuel, glucose comes in, it gets split, we make a couple of ATPs, that part happens without oxygen. But then to get the 30-some ATP from each glucose molecule, we absolutely require the oxygen. That gets us a whole lot of ATP, and let's see what we can do with ATP. We can pay for pumps. Importantly, the sodium-potassium pump. All of our cells have them, and they're studied all over. Here's just one, but they're all over the cell membrane. Sodium gets pumped out of the cell. Potassium gets pumped in. There's already a lot of sodium out of the cell and a lot of potassium inside, but we push more. We make sure that we keep pushing those. And we'll see later in the semester, we need it. Well, you actually, let me remind you what we need it for. You may recall muscle contraction requires changing levels of sodium and potassium, so do neuron firings. So it's really important that we maintain the appropriate ion levels inside and outside of the cell. And to do it, we have to pay with ATP every single time. Just like the sodium potassium pump, we also have a calcium pump that requires payment each time we pump calcium ions. I'll remind you, calcium ions are used in a muscular contraction. They're absolutely required inside the cell for muscular contraction. But the calcium doesn't stay in the cell. It gets pumped out. And in general, having too much calcium, even in muscle cells, is detrimental to the cell. And so it's really important that these calcium ion pumps be functional and be able to empty the cell of large amounts of calcium. And again, in class, we'll talk about what the consequences are when we don't have enough ATP to pay for this. Other jobs that get paid for with ATP, transcription and translation. They're pretty costly. We need to pay for those. We need energy to pay for those. And making the proteins and what these proteins are for, some of them are needed to make the cell itself, the structure of the cell, the cytoskeleton, et cetera. But some of it just gets damaged and you just have to fix damage to the cell. Well, that's going to cost something. And so you have to pay for it with ATP. And if you don't have enough ATP, you can't do it. And one more thing. Cells can make lipids, but there's one kind of cell in particular that's most famous for this, the cells in the liver. One of the many, many jobs that those cells do is to make lipids. But then those lipids get stored in adipose tissue somewhere else in the body, or they're going to get used for fuel in the other cells. And so it needs to get transported from the liver somewhere else. You may recall lipids, the thing that all the lipids have in common is that they don't dissolve in water. And yet we're going to try to ship them through channels that are largely filled with water. And so the way that the lipid can get around is by being attached to proteins. The proteins can dissolve in the water and able to shuttle the lipids around. You have to make those proteins. And then on top of that, you have this complex, this lipid transporter protein complex that has to be let out of the cell through exocytosis. It needs to get out from the inside of the cell to the extracellular fluid. That costs energy in the form of ATP. Let's see. So before we get into the electron transport chain, we go through what's called the citric acid cycle. You may have learned it with that name, or Krebs cycle is the other term that's used for it. We're not going to go through all the details. What I want to remind you is an energy-rich molecule comes in. Those of you who know lots of detail, you take a glucose, remember, break it up into 2 pyruvates, and then the pyruvate gets converted to acetyl-CoA, and then that goes through this cycle. What matters here, what I want you to remember, is that in doing this cycle, we produce what are called electron carriers. They take these high energy electrons to the electron transport chain. So there's NADH and FADH2, which is somewhere, I don't see it. Anyway, you make some electron carriers, and those electron carriers take the electrons to the electron transport chain in the mitochondria, and that's where we're going next. So this is the inner membrane of a mitochondrion. It has these embedded proteins. Hey, we had to make those proteins using ribosomes. But anyway, we got some proteins that are embedded here in this membrane. The electron carrier, NADH, is going to drop off some electrons. Those get handed down and handed down and handed down. Some protons are getting pumped across the membrane. You may recall this. And then there's two things that are important to note here. One is what happens to the electrons at the end of this electron transport chain? They get put onto an oxygen. That, my friends, is the secret of what oxygen is all about. That is the job, the only job that oxygen has in our bodies, is to accept the electrons here at the end of the electron transport chain and take it away in the form of, well, in the form of water. But what's important is the oxygen has to accept these electrons. In the absence of the oxygen, the electrons stay put. This whole thing grinds to a halt. We can't transfer protons across this membrane. And so this final step where we let the protons come pouring through the ATP synthase, that beautiful protein that spins and makes a bunch of ATP. Oh, it's so wonderful. So this is the secret. This is how we get all that ATP, is using this protein ATP synthase that's powered by having these protons coming through. So in the absence of oxygen, the electrons don't flow, so the protons don't flow, so the ATP doesn't get made. And that is the tragedy of going without oxygen. And we'll see this in more detail in class when we talk about hypoxic injury, having low oxygen levels for the tissues and all the damage that can result from not having oxygen to accept these electrons. So we will cover those accidents and problems that occur due to those oxygen deprivation accidents, and we'll do that in class. See you then.